Hey everybody, welcome back to Neuroscience. Today we're going to go in depth of the visual path and vision itself. The visual path is pretty straightforward. It goes from the retina to the visual cortex through the striate area. And most of the axons will pass through the lateral geniculate body of the thalamus. So all of this is going to be responsible for vision interpretation and the act of accommodation. So because of the accommodation, there is some fibers that pass through the superior colliculus and the Edinger-Westphal nuclei. So that's going to be responsible for the pupillary reflex. So there it is. It's responsible for the parasympathetic innervation of the pupil. Lastly, there's a bunch of fibers that will pass posteriorly to make the genicocalcarine tracts. Okay, so in that tract, there is the radiation when the lateral geniculate body passes through the retrolentiform part of the internal capsule, and then that ends into the occipital lobe. So the lateral geniculate body is made out of 6 nuclei, and they're divided in the magnocellular and the parvocellular. So the nuclei in pink, so 2, 3, and 5 are going to be responsible for ipsilateral vision, while 1, 4, and 6 are going to be responsible for contralateral vision. And that's due to the decussation, remember. The Meyer's loop is where the fibers from the lateral geniculate body loop over the lateral ventricle, like right here, and it goes to the occipital lobe. The lateral geniculate body, once again, has all these fibers, these nuclei, and they end in areas 17, 18, and 19 of the occipital lobe or pole. Okay, so the medial fibers of the visual cortex is going to carry the lower contralateral visual information from the superior retina, and that will go to the cuneus gyrus. So what does that mean, right? So it's going to carry the opposite end of the vision, like so. If let's say this is like a full pie, if the medial fibers of like the right eye, right, is going to carry information from this side of the field, and it's going to be the lower contralateral. So like it's going to be the lower fourth of this like imaginary pie. The lateral fibers, on the other hand, will carry the upper contralateral information, and that will come from the lower retina, and that will go into the insula. So the lateral fibers are going to carry the information from the upper, like we're talking about the same eye, right eye. It's going to come from the upper field of vision, right? So it's going to cross and then hit like the lower retina. So all of this is going to be linked to the memory, because this is how we store visual memory in the cortex. This is where the interpretation occurs. Okay, so what is the visual field? The visual field is the area that can be seen when the eye is steadily gazing like straight ahead. And you are able to interpret static objects, so like a tree, kinetic objects, so if the tree is moving because of the breeze, color, and form. So what's important to know is the temporal field is the one that crosses over. The nasal field is going to be carried by one tract, so there's no decussation. So this is the nasal field, because this is like medial vision, but it's going to be carried by the temporal retina. So this is the decussation. I'm sorry, not decussation. Nasal is going to go like straight here. Decussation is from the lateral field. So the lateral geniculate body is going to get fibers from the temporal field. So over here, that's the information. It's going to cross. So this is going to be the decussation right there. The lower quadrant of vision, so like here, is going to go to the ventral part and goes to 2/3 of the lateral geniculate body. while the macula vision, so like very center vision, is going to go to the dorsal third of the lateral geniculate body. The upper quadrant of the retina is going to get images from the lower visual field. So this is a little bit tough to like kind of figure out. I really recommend kind of drawing out the field and which goes which, but when in doubt, everything crosses over except for the medial tract, so the nasal vision. Okay, so there's a bunch of different kinds of blindness or visual field defects. Only true blindness, total blindness, is going to be the injury of the optic nerve itself. And it can be either in one eye, the other eye, or both eyes, depending on where the lesion is. So if the lesion is here, there's going to be monocular vision loss. The entire optic tract is damaged. This eye is going to be blind. So in bitemporal heteronymous hemianopia is going to be a problem with the chiasm. So there's going to be problems with decussation. Remember, lateral fields cross over. If the lateral fields are gone, you're going to have something called tunnel vision. So it's going to be black on the periphery, but your center vision is going to be okay. Usually caused by some kind of pituitary tumor compressing on the chiasm. The unilateral nasal hemianopia is going to be damage to the lateral part of the optic chiasm. So that is going to cause like contralateral nimbus hemianopia. So it's going to be like the left side of your vision is going to be gone. Usually caused by some kind of aneurysm of the internal carotid artery. So the right or left homonymous hemianopia is going to be complete destruction of the optic tract or the geniculocalcarine tract, and it's going to cause the complete blindness in the opposite half of the field of vision, so like here. There's a bunch of other visual field defects as well, like contralateral superior quadrantopia. So this is kind of like pie in the sky. There's going to be like a dark spot in the sky in the upper left quadrant. If the Myers loop is damaged, there is something called like a hole in the ground if the like part of the left geniculate nuclei radiation is damaged, like right here. And then there can be contralateral hemianopsia with macular sparing if the lesion is in the optic in the optic region itself, or if the posterior cerebral artery is occluded. Okay, so once again, homonymous hemianopia is going to be blindness in half of the field of vision with macular sparing. Right there, it's actually the occipital lobe itself being damaged because of the PICA artery. So contralateral inferior quadrantic anopia. So that is going to be our pie in the sky. That can be caused by destruction of the upper lip of the calcarine sulcus or the parietal fibers on one side. So that's going to cause visual loss in the lower quadrant of the opposite field of vision, so that would be the hole in the ground. Okay, so sorry, the contralateral inferior, that's the hole in the ground. Contralateral superior, this is going to be pie in the sky. And this can be caused by damage of the lower lip of the calcarine sulcus or the temporal Myers loop, so like around here. Temporal lobe lesions involving the Myers loop is going to cause a pie in the sky, and lesions anterior to the lateral geniculate body, especially the Edinger-Westphal nucleus, is going to cause the pupillary light reflex problems. Okay, so what is the pupillary light reflex path, right? It's a parasympathetic innervation path, and this is when the pupil is going to be constricted in order to protect the retina when light intensity is too high. So the light hits the eye, and then it will stimulate the ganglionic cells. Action potential goes like down, and this is going to be either decussation, or it's going to go straight from the medial vision, and it hits the lateral geniculate body. And then from there it goes to the occipital lobe. Occipital lobe, it processes the information and sends it back to the Edinger-Westphal nucleus. And then from there it's going to send the action potential to the ciliary ganglion, and then to the ocular motor nerve in order to blink. So the Edinger-Westphal nucleus will also relay the information to the ciliary muscles. So there's a phenomenon called consensual constriction. So if light is going to be shown in one eye, there's also a constriction of the other eye's pupil, even though light may not be shown in the other eye. Okay, so the blink reflex is also consensual. We test this by something touching the eye, or like a puff of air, like those horrible machines that you have to get tested at the eye doctors. And the little puff of air is going to put a little pressure on the cornea, and then the trigeminal 1 afferent in the cornea is going to transmit the information to cranial nerve number 5, and then from there it's going to go to the ganglionic root and then the spinal trigeminal nucleus. From there, the information is processed and then sent to the facial nucleus, which goes to the abducens nucleus, then to the facial nerve, and then all the way back to the eyes, and either causes the eye to blink because it innervates the orbicularis oculi, or in some cases it makes the lacrimal gland get activated. So all of this is to protect the eye from debris and trauma. Okay, so stereopsis is our ability to maintain depth, because it's a binocular vision phenomenon. So we're able to perceive depth and visual texture because of the minute details, right? Another phenomenon is visual attention. So this is the process or the act of directing and sustaining the eye towards a visual stimulus, causing the image of the stimulus to be centered onto the fovea. So fovea has the sharpest vision, so we want to like focus on that. Like our attention is going to be focused on the clearest vision. And that's useful in tracking, because tracking is the dynamic movement of the eyes to keep the image of the object continuously on the fovea. Saccades is the rapid change in fixation from one point to another, and perception occurs at the moments of fixation. Interestingly enough, tracking perception is continuous, so you're kind of like aware of what's moving and what is not moving. So like if you're tracking like a bird flying, you know that the bird is flying, but the clouds in the skies is still. Saccades is more like you kind of you switch attention from bird to cloud like quickly, and the perception is going to occur like when you're focused on the bird, the clouds aren't moving. When you're focused on the clouds, you know the bird is moving. Voluntary saccades is going to be generated by the frontal eye fields, the superior colliculi, and the parietal cortex. The stimulus-generated saccades is going to be generated by additional input from the occipital lobe. So for the voluntary saccades, like back with the bird in the cloud, you are actively switching from bird to cloud. The stimulus-generated saccades is when you're a bit startled. So like another bird shows up, or better yet, like a mosquito shows up in your vision, and all of a sudden you switch over. Because of the it's not just input from the occipital lobe, but like from the other senses, right? So once that catches your attention, like automatically the body switches attention. Okay, so convergence is basically the use of the extraocular muscles to like focus the eyes on a midpoint where you can fixate the target. And something called a convergence near point, that's the location where there's max convergence. So it can't be too far, and it can't be too close. We test the convergence by like asking the patient to look at your finger as you move it back and forth, right? Like about like 3 inches from the face is when the eyes start converging, so they look cross-eyed. That is going to be like past your point of max convergence. This is when like the vision starts getting blurry. Visual scanning, back to this, is a process. It is a very dynamic process because it's the integration of the 3 subcomponents of pursuits, saccades, and fixation. And all of that plays in a role in visual perception. So you have to have the capacity to interpret the visual stimuli, recognize the similarities and difference, and assign meaning to the sight. Okay, that's it for now. Thank you for your attention and good luck studying.